Does he like me, or does he like me? Well, might as well find out when I ask him.
Hey there, son. Guess who's here to see you?
Who?
Grandpa.
I hate Grandpa. He always pinches my cheek.
Well, he's really excited. And I think he's got a present for you.
Hey, Grandpa.
Hey, how are you doing?
Okay.
Well, I have something for you.
What is it? It's a book.
Yes, it is. Open it up. See, this book, my father read it to me, I read it to your father, and now I'll read it to you.
I hope it's not about sports, because I like kissing and ponies.
Oh, there's plenty of kissing in this book. Alright. Today, even school children have heard of DNA, and scientists routinely manipulate DNA in the laboratory and use it to change the heritable characteristics of cells. Early in the 20th century, however, the identification of the molecules of inheritance loomed as a major challenge to biologists.
My God, Watson! What's that sound?
Nothing.
Stop arguing with me, Watson!
Are you trying to imply like I'm Watson, like in Sherlock Holmes? I'm not Watson, you're my sidekick, you're the Watson here.
No, I'm not!
Stop arguing! Crick, we just should do what we came here to do!
God, what is the genetic material?
I don't know, it could either be DNA, or it could be protein.
Well, let's look back at some of the other experiments that have been done and see if we can find, maybe, what it is.
That's a brilliant idea, Watson!
Where do they go, those
That's a brilliant idea!
Let's take a journey throughout the land so we can get an idea as to what the genetic material is.
When the moon hits the sky like a big pizza pie, that's amore.
Two scientists named Hershey and Chase devised a very clever experiment to find out what the genetic material was. It was either protein or DNA.
This chocolate chase is delicious. Why couldn't it have been my chocolate?
Hello, Hershey. Hello, Chase.
Hello.
Hello. We heard you devise an experiment to find out what the genetic replication material is.
That we did. Well,
Could you explain it to us?
yes. It all started when we...
Mark the DNA phages with radioactive phosphorus-32. We then infected the E. coli we used with the phages. Then, we removed the protein parts from the infected cells with the centrifuge and found that the radioactive marker showed up only in the bacterial cells and not the protein parts, which hinted protein may not be the genetic material. Chase, describe our second experiment.
In our second experiment, we traced the phages with radioactive sulfur-35, which is not found in DNA. After separation, the radioactive marker then was found in the protein parts, but not in the infected bacteria, thus confirming that the genetic material which infected the bacteria was DNA.
Wait, Grandpa. I thought there was kissing in this book.
Oh, you just wait! Next, Watson went to visit Rosalind Franklin, who had been doing lots of research on the structure of DNA molecules.
Wait, I think we're here.
This is kind of an old laboratory. Oh, hey, Rosalind Franklin.
Hi, Dr. Watson.
I heard you're doing some interesting research into the structure of DNA molecules.
Oh, you mean my X-ray diffractions? Yeah, they're right over there if you want to take a look.
Oh, all right. Oh, these are pretty interesting.
Documents lifted.
What are you doing? My life's work!
Look at this research that I snagged from Rosalind Franklin.
Just take it back from her laboratory?
More like a kitchen. Anyways, this seems to suggest that the model of DNA is in a double helix formation. Take a look at this, Watson.
Don't call me Watson, Watson! Yes, this does look like a very helpful piece of information. Maybe with this we can finally put our structure model together. But I think first we ought to do some more research.
That's a great idea! Tally-ho!
I think I've got it.
What is it?
The DNA is a double helix structure with a phosphate backbone on the outside made of deoxyribose sugars and phosphates. And then on the inside of the helix must be nitrogenous bases, 4 of them: adenine, thymine, guanine, and cytosine. The adenine will always bind with the thymine, and the guanine will always bind with the cytosine. So, in every DNA molecule, they're equal.
That makes so much sense! Watson, I think we've got it! Is there anything else?
Well, I think that within the nitrogenous bases, there are 2 types: pyrimidines and purines. The purines are adenine and guanine, and the pyrimidines are thymine and cytosine. See, if you put 2 pyrimidines together, then the DNA will be far too narrow. But if you put 2 purines together, it'll be far too wide. That is why a pyrimidine always has to bind with a purine. Adenine, being a purine, will always bond with thymine, which is a purine, or which is a pyrimidine. But the guanine, which is another purine, will always bond with cytosine, a pyrimidine. That is why DNA is always equal in length.
That makes so much sense now. It all fits together. Now all we have to do is find out how it replicates. Let's go on a journey. With all of this knowledge, Watson and Crick wrote a paper that eventually won them the Nobel Prize. However, they and many other scientists were still curious about the replication of DNA.
Do you hear that beast? Why, I do. What could it be?
I don't want to find out. Mamma mia, here I go again.
Let's go on a journey.
Don't know the words. I don't know either. Rockets in flight. Afternoon delight. Afternoon delight.
Watson, why are we here?
I heard from somebody that we could learn about replication here.
Who goes there?
Startled us. Sorry, I'm Crick.
I'm Watson.
Oh, I've heard that you boys seek the origin of replication. Well, replication is a tricky business. There are several enzymes you'll need to be familiar with. DNA helicase unwinds the parent strand and separates it into 2 distinct strands. RNA primase then lays down an RNA primer on each strand, whereby DNA polymerase can attach and create a new daughter strand. Remember, DNA polymerase always reads from the 3' to 5' end. Remember, boys and girls, DNA always separates into 2 distinct strands, the leading and lagging strand. On the leading strand, RNA primase only lays down 1 piece of RNA primer, and DNA polymerase builds a new strand from start to finish. On the lagging strand, however, DNA polymerase has trouble reading from 5' to 3'. Therefore, RNA primase must lay down multiple primer fragments. DNA polymerase can then lay down new strands starting with each of these primer fragments. These result in different sections of DNA known as Okazaki fragments. Once this is all done, ligase comes in and finishes the job, joining the phosphate backbone fragments together to create a continuous strand.
Wow, thanks.
Now that that's all finished, would you boys like to join me in my hot tub for some brain soup and perhaps a wine cooler?
No thank you, but thank you.
Good day.
Oh, finally, we've done it, Watson.
We're finished, Watson! That's a brilliant idea!
I hope it's not about sports, because I like kissing and ponies.
Why couldn't it have been my chocolate?
Go home.